synapses. 24. Myelination of axons in the peripheral nervous system is carried out by Schwann cells, insulates the axons to reduce current leakage, increases the speed of action potential propagation through saltatory conduction, and conserves energy because the active excitations are restricted to the nodes of Ranvier. 25. Channel-rhodopsins are light-responsive cation channels that can be engineered to be expressed in specific neural cell types. Opening of these channels in response to light allows sodium ions to leak into the cells, raising the membrane potential to the point when voltage-gated Na+ channels open and further depolarize the membrane in an action potential. 26. The powerful patch clamp technique can be used to record ion currents through a single ion channel. Using this technique, individual gated channels are observed to randomly flip between open and closed in an all-or-none fashion to generate the aggregate membrane current. Such experiments have also shown that even cells that are not electrically excitable have a variety of gated ion channels. 27. Synaptogmon 1 is activated by binding to calcium, which flows into the cell through voltage-gated Ca2+ channels upon the arrival of an action potential. This activation drives neurotransmitter release. 28. Based in part on their firing pattern, neurons can be categorized into functionally different types. The firing properties of each neuron type are largely determined by the dynamic levels of depolarizing Na+ and Ca2+, and hyperpolarizing K+ channels, and are under homeostatic control. 29. Excitatory neurotransmitters open cation channels leading to sodium or calcium influx and postsynaptic membrane depolarization, whereas inhibitory neurotransmitters open potassium or chloride channels, suppressing firing by making depolarization harder. 30. Astroglial cells in the brain can actively import the amino acid glutamate from the synaptic cleft, convert it to the amino acid glutamine, and release the glutamine into the cleft to be subsequently taken up by the presynaptic neurons. The neurons then convert it back to glutamate. This function of glial cells is important because glutamate is an excitatory neurotransmitter and should be cleared from the synaptic cleft rapidly after its release. 31. Neuromuscular transmission involves the sequential activation of several ion channels. The events sorted to reflect the sequence leading to muscle contraction are as follows: opening of voltage-gated Ca2+ channels in the motor neuron, opening of ionotropic acetylcholine receptors on the muscle cell plasma membrane, opening of voltage-gated Na+ channels in the muscle cell plasma membrane, opening of voltage-gated Ca2+ channels in the T-tubules, opening of Ca2+ release channels in the sarcoplasmic reticulum membrane. 32. Dopamine is a neurotransmitter involved in the reward pathways in the brain, and its decreased activity has been associated with diseases such as Parkinson's disease and attention deficit hyperactivity disorder, ADHD. Methylphenidate, a dopamine reuptake inhibitor. Amphetamine, a dopamine-releasing agent that triggers the release of dopamine into the synaptic cleft. Forskolin, a sensitizer of dopamine receptors. And carbidopa, which enhances the availability of the dopamine synthesis precursors in the brain, are all likely candidates to treat these diseases. Chlorpromazine, a dopamine antagonist that binds to and inhibits dopamine receptors, however, is likely not a candidate to treat these diseases. 33. The inactivation rate of voltage-gated Na+ and K+ channels can regulate the firing frequency of neurons. To support high firing rates, both channels should rapidly inactivate. In other words, the combination of rapid inactivation of Na+ channels with rapid inactivation K+ channels results in the highest firing frequency. 34. Long-term potentiation and long-term depression share a similar scheme, but result in opposing effects largely due to the different levels of Ca2+ that they bring about. Study Guide. Chapter 15. Cell Signaling. 1. Paracrine signaling acts on neighboring cells via local mediators. 2. Second messengers are small chemicals generated in large amounts in response to receptor activation in signaling pathways and diffuse away from their source to spread the signal. Some are water-soluble, for example calcium ions and cyclic adenosine monophosphate, CAB, and some are lipid-soluble, for example diacylglycerol, DAG, and inositol trisphosphate, IP3. 3. Activation of an upstream guanine nucleotide exchange factor, GEF, activates a GT-POSA by facilitating its conversion into a GTP-bound form through guanine nucleotide exchange. 4. Negative feedback generally dampens a cellular response, while positive feedback loops and cooperative activation sharpen it. 5. For the Greek letter alpha subunit of a trimeric G protein, a G protein coupled receptor, GPCR, acts as a guanine nucleotide exchange factor, GEF, whereas a regulator of G protein signaling, RGS, can act as a GT-POS-A activating protein, GAP. 6. While cholera toxin causes constitutive G's activation, pertussis toxin prevents G activation. Both of these result in a net activation of adenylyl cyclase, leading to higher CAP levels. Note that adenylyl cyclase is not the only target of G. 7. Stimulation of protein kinase C, PKC. Activity near the plasma membrane results from elevated cytosolic Ca2+, induced by inositol 1,4,5-trisphosphate, IP3, production, and from binding to diacylglycerol and negatively charged phospholipids. IP3 and diacylglycerol are second messengers in the cytosol and plasma membrane, respectively, and are produced from phosphatidylinositol 4,5-bisphosphate by the action of BLK. 8. Following the phosphorylation of the receptor by rhodopsin kinase, aristin binds to it to prevent transducin activation. Other mechanisms, such as binding of RGS, regulator of G protein signaling, to the G protein Greek letter alpha subunit or activation of guanylyl cyclase, are also involved in visual adaptation in rod cells. 9. Consider visual transduction in rod photoreceptors in the vertebrate retina. Each of the following steps can amplify the signal in this pathway: activation of transducin by active rhodopsin, blockage of Na plus influx by cation channel closure, and depletion of cGMP by active cGMP phosphodiesterase. 10. Mice or humans with defects in RK, or its downstream regulator aristin, have a prolonged light response, incapable of adapting quickly to sudden drops in ambient light intensity. 11. Unlike insulin receptor, IR, activation, epidermal growth factor receptor, EGFR, activation involves an asymmetric arrangement of the intracellular kinase domains. 12. Consider a signaling protein that is only made up of one SH2 domain and two SH3 domains. This protein is most likely an adapter protein. 13. Activation of ROS-GEF is the major way by which the monomeric G protein ROS is activated in receptor tyrosine kinase signaling. 14. Scaffold proteins help prevent crosstalk and improve precision in signaling pathways, but they also limit diffusion of pathway components and thereby reduce the opportunities for amplification and spreading of the signal to different parts of the cell. 15. The plasma membrane-bound Pi3 kinase principally phosphorylates inositol phospholipids rather than proteins. It can be activated downstream of receptor tyrosine kinases or G protein-coupled receptors, and generates docking sites for various signaling proteins. PTEN phosphatase counteracts Pi3 kinase by dephosphorylating the 3' position of the inositol ring. 16. Mainly functioning in protein-protein interaction, pH domains are found in many signaling proteins, including plextrin, AKT, PDK1, and SOS. 17. Proteins of the SRC family, for example SRC, FUN, LCK, and HCK, have important roles in several signaling pathways in mammals. A typical SRC family protein is a cytosolic tyrosine kinase, contains SH2 and SH3 domains in addition to a kinase domain, has covalently attached lipid chains, and is located on the cytoplasmic side of the plasma membrane. 18. The SH2 domain of STAT proteins plays a key role in cytokine receptor signaling. This domain can bind to phosphatidylserine on an activated receptor molecule, or on another STAT molecule in a dimer. 19. Activated KF receptors can be endocytosed by two distinct routes. One is dependent on clathrin-coated vesicles and leads to further activation, whereas the other one depends on caveolae and leads to inactivation. Inhibiting the formation of caveolae can therefore activate, rather than inhibit KF signaling. 20. JAG-delta ligand binding to Notch receptor releases the intracellular domain of Notch, which enters the nucleus and converts a transcriptional repressor into a transcriptional activator, turning on the expression of Notch target genes. 21. WNT binding to frizzled and LRP results in the disassembly of the Greek letter beta-catenin degradation complex, containing APC, axin, CK1, and GSK3, which allows Greek letter beta-catenin to accumulate and enter the nucleus, where it binds to LEF1/TCF, displacing the co-repressor GRUCHO. 22. Both C and Greek letter beta-catenin are phosphorylated by GSK3 and CK1 in the absence of stimulation in hedgehog and WNT signaling pathways, respectively. They are both ubiquitinated and undergo proteolysis in the absence of signal. Greek letter beta-catenin is stabilized and can enter the nucleus only in the presence of WNT. C enters the nucleus in the presence of hedgehog and acts as a transcriptional activator, but its fragment, produced from partial proteolysis, can also enter the nucleus and act as a transcriptional repressor in the absence of signal. 23. All nuclear receptors have ligand binding and DNA binding domains, and can directly bind to DNA. 24. Ethylene receptors in plant cells have a copper-containing ethylene binding domain. 25. In the presence of a lateral light, auxin efflux transporter proteins become primarily localized to the side of the cell that is away from the light source. This redirects the auxin flux mainly to the darker part of the growing shoot, where it stimulates the elongation of the cells, in contrast to its inhibitory effect on the elongation of root epidermal cells seen in gravidotropism, bending the shoot towards the light source.